Hello, I'm going to explain the central dogma of protein synthesis or of organisms or however you want to say it. So, central dogma is how we get from the DNA to um, mRNA to our amino acids, which form a polypeptide chain, which form protein, and protein forms organisms, so us. So, this screen is transcription. Right up here's our RNA polymerase. Here's our DNA strand. Our top is our coding strand. Bottom is the template strand. RNA polymerase reads the template strand and somewhat reflects the coding strand, except instead of thymine, it pumps out uracil because it's ribonucleic acid, not deoxyribonucleic acid. So, boom, here's our helicase. It's going to come in and unzip our DNA, break all these H bonds. Our RNA polymerase runs along the DNA until it hits the promoter region. When it hits the promoter region, it's going to begin transcribing what it reads on the template strand, and it's going to form mRNA. It takes in these nucleotides that are free, and it's going to use them to create the mRNA. And RNA polymerase is going to get cut off when it hits the termination or the terminal uh, region, and this is going to, it basically represents a gene that the nucleus wants coded for. So this mRNA is going to be transcribed for a specific gene. Boom. So this mRNA is going to come down and it's going to get out of here. It's out of the nucleus and it's going to go to a ribosome. And then down here on the bottom is our finished product of DNA. It's all good. It got zipped back up. Um, yeah. Also, RNA polymerase reads 3' end to 5' end and transcribes 5' to 3'. So it's just like the corresponding way. Yeah. All right. For translation, this funky looking thing is our ribosome. It has two subunits. The bottom is the small subunit, top is the large subunit. This black strand is our mRNA. These are tRNA, which are transfer RNA. Think of these like a taxi for amino acids. Um, tRNA has an anti-codon end, which binds to its corresponding codon end on the mRNA, and it has an amino acid attachment site end. And this is where it's going to bind to its corresponding amino acid. These tRNAs have specific anticodons and amino acids that they attach to. These are specific, like these all go together. So here's the A site. It's where the, so to start, the codon AUG, which codes for the amino acid methionine, methionine is going to come in and it's going to come right. And then after the A site, it's going to move on to the P site. This is the peptide site. This is where the tRNA comes down and attaches its anticodons, which correspond and are the correct opposites of the codons, the nucleotides on the codon of mRNA. From here, our methionine is going to start the polypeptide chain. Ignore this. This is like later in the process. We'll go over that in a sec. So it's going to put down the first amino acid, and it's going to move to the exit site, and it's going to fly away because we don't need him. He's probably going to go get another amino acid. And it's going to be methionine again because he goes for only one type. Okay, and then boom. So that's after methionine. More tRNAs are going to come in, and they're going to like bind to their specific or their corresponding codon. And it's going to create this polypeptide chain, which keeps building, and they're formed with peptide bonds, and it's going to create a protein. So I'm going to hurry up, and my 5 minutes is short, and so my disease or my protein synthesis related disease is going to be um, Huntington's disease. Basically, this is where the the DNA strand has a bunch of CAG nucleotides in a row, so codons in a row, CAG codons, and this calls for this forms glutamine, the amino acid glutamine, And RNA polymerase is going to read these. And sometimes, if there's like 35 or more, which happens in Huntington's disease, it's going to start messing up. And this can cause these mRNAs to be like false or bad, and then boom, they still get turned into a polypeptide, which turns into a protein, which goes to the brain, and a lot of times it causes nerve cell death in the brain because of a faulty structure for many different reasons. Um, and some symptoms of Huntington's disease is like involuntary movements, being off balance, uh, bad hand-eye coordination, a bad mood. So this is common with like older people, and like kind of corresponds with Alzheimer's and stuff. They can have mood swings, and then cognitive, like also, which would also be like thinking and just understanding topics. All right, that's crazy. 6 minutes. All right, bye.